Hello everyone, let's start again in this cell signaling course. We in the last two classes have seen that what is signaling, why it is required, and what are the need of the signaling in terms of what signaling needs and why we need signaling. Now let's see the different types of signaling which is prevalent in the living world, and more of examples we will take in this particular lecture is from the animal world. So when we talk about the type of signaling, the very first thing which is very relevant to the title itself, the cell signaling, is the subcellular signaling or intracellular signaling. So what we understand from subcellular or intracellular signaling? We know that a cell has multiple compartments, subcellular organelles. And the existence or survival of the cell is because of the efficient communication between these organelles. So there must be a communication between these organelles, and we have seen in the previous classes that how communication is the simplest form of signaling, right? So whether it is endoplasmic reticulum, whether it is mitochondria, whether it is nucleus or lysosome, you think about any of those subcellular structures. Their activity, their function, their efficiency in performing their function, all that is interrelated and probably interregulated as well. So those regulations via the signaling is basically subcellular or intracellular signaling. Now when we think about specific subcellular organelles, it is clear that mitochondria is powerhouse, right? I guess everybody knows this, appreciates this, because this is the sole place where bulk of my ATP is generated, right? So the signal to generate ATP is very well there in mitochondria, communicated also by virtue of presence of specific proteins on its surface. Also, under certain cases, mitochondria can give the signal for apoptosis. I hope you understand the term apoptosis, which means that the death which is happening, it's a programmed death. When we say programmed death, what does that mean? That there must be a signal which will communicate to the cell through mitochondria. That cell has to die for whatever reason, for whatever circumstances it is facing. Now then there are structures which are called Golgi, and we know that from here a lot of secretory vesicles are produced, and these vesicles reach to their destination by marking. Each vesicle is marked and it goes to a particular location, particularly the ones which are targeted for delivery into the lysosome. So Golgi is communicating with lysosome by sending specific proteins to the lysosomes. Then ER, which communicates with Golgi and mitochondria to ensure that protein homeostasis is maintained, secretory products are being made, and the membrane potential of the mitochondria is maintained. More importantly, the cell division, it is regulated by internal signaling. In the previous class, we discussed that how proliferation, whether it is mitotic division or meiotic division, both are tightly regulated by signaling. So when a cell decides that it has to undergo division, often into the cells it comes from internal signaling, which means that you weigh out the presence of different organelles, components, and the potential of the cells to divide, and it undergoes the division. So under subcellular or intracellular signaling, we can understand that different components of the cell is communicating, signaling, so that efficient functioning of each of those components is maintained and cell is healthy and surviving fine. The other type of signaling which we can understand, particularly for the cells which are involved in the process of secretion, where I have said that it is self-signaling or autocrine, which means that the secretion which is being produced by the cells works on themselves, and when they work on themselves, they work sometimes through this feedback mechanism. So in case of autocrine or self-signaling, the source cell is same as the target cell. When we talk about same, basically what is happening here, if suppose this is a cell which produces signal in form of these compounds which are secreted outside. Now they will work on itself, they should find a receptor. When they bind to this receptor, they give signal to self saying that more of these molecules needs to be produced or not. So it's a self-signaling. Also in the previous class I mentioned about the quorum sensing, if you remember, and I told you that this is one way by which the bacteria forms the film. Also there is a phenomena which is called bioluminescence, where these organisms, they produce light. When they are enough in number and they cross the threshold, the signaling actually helps them produce light. So if you consider a scenario where one cell, such a cell is, or multiple cells which are present in the system, they are releasing signals around them, they are releasing the signals, and these signals are working on one of them only, basically same, right? So in this case what happens, if the signal amount is less because the number of cells is less, you would not see the phenomena of bioluminescence because the signal which is produced by them does not cross the threshold. Now consider a scenario where suddenly you have huge amount of signal which is present around them. So when you have huge amount of signal present around them, they respond to that signal and start giving this light out, and you see the bioluminescence. So where does this more signal come from? Either the cells start to produce more signal, or there are more cells to produce more signal, right? So the self-signaling not only helps through control the function of the cell by feedback mechanism, but it also helps in one of these quorum sensing event which we saw in the previous case, in the biofilm formation as well as the bioluminescence which is happening in the bacterial system. Now one of the other signaling type is called contact-mediated signaling. The name itself is very clear. It suggests that the two entities which are participating in the signaling, they must come in close contact. In the previous cases, whatever we saw, you would have noticed that cell is producing the signal and a cell which is far off could actually receive the signal, right? So the two cells which are signaling or part of the signaling event, they are not coming in close proximity. But in case of contact dependent signaling, what exactly happens? That if you consider a scenario here where this is a source cell and this is the target cell. You remember we said that there is a source and there is a sink. So basically source cell is the source of the signal and target cell is the sink for that signal. So since it is a contact dependent signaling, what you can simply assume that the signal is not being released out. Rather, the signal is also membrane bound. And the receptor which is recognizing it is definitely membrane bound. Okay. So the two cells which are participating in the signaling, their signal and the receptors both are membrane bound, which forces them to come together and engage in the signaling process, right? So basically in the contact mediated signaling, contact dependent signaling, you have both signal and receptor which are membrane bound, which forces them to come together and be in the close proximity, right? So these two cell types which will be engaging in contact dependent signaling will show some distinct feature which we will discuss later. However, one thing which I would like to emphasize, normally whenever we will be talking about the signaling process and when we will be talking about target cell, we by default understand that this is the receiver. Target cell is the receiver, and one of the characteristics of the target cell would be to have the receptor. Okay, so target cell is the receiver and it has the receptor. So basically when we talk about cell signaling, the cell which carries the receptor is showing the consequences of signaling. So this will be a signaling cell. Now we will see some cases where, so what you can imagine, that under normal circumstance, out of the two cells which are involved in signaling, always the target cell is doing bulk of signaling by virtue of having the presence of the receptor. But in some cases, we will see later that both cells which are part of this contact dependent signaling are simultaneously undergoing the signaling and showing the consequences of signaling. Normally, when a cell which is producing the signal, or the transmitter cell or the source cell which is located little far away from the target cell, only the target cell bearing the receptor shows the signaling. Now if we think about the other type of signaling which is present in the living world, you can think about the signaling which is mediated by the messenger. Here messenger is nothing else but the signal or ligand, or maybe, which I told early, stimulus. What it means that your signaling is going on with the help of this messenger. So you might wonder that what is different? All that you have told until now about the signaling requires the signal or the messenger. Now what I want to emphasize here is that how far the messenger is traveling. If the messenger is traveling to a shorter distance, it is called autocrine signaling or short range signaling. Rather, I should say, when the signal or the messenger travels to a short distance, it presents the example of short range signaling, which is often seen in the autocrine signaling or in exocrine signaling. If you remember, your exocrine glands, they produce secretion and they locally deposit that secretion. The cells which are around that area respond to that. Other way is where you can have a messenger traveling to a long distance, and we see this long range signaling as exemplified in paracrine signaling, which is also a classic example of how endocrine system works. So what you would notice in case of short range signaling, that you will have a target cell which will be producing the signal. This signal will bind to the receptor and signaling will go on here. So from previous slide, if you remember, I said the cell which has the receptor ends up signaling. Also in this context of this autocrine signaling or short range signaling, you have this feedback regulation. What I do not mean from here is that other signaling do not have feedback regulation. Here you can clearly see that, right? So this is short range signaling, where the signal produced by a cell is consumed in the proximity only. It could be working on the self, or it could be working on a different cell which is very close by. But in case of the endocrine signaling, I am sure everybody in this class is aware of the endocrine system. Most of the hormones which are produced in our body is from the glands which are called endocrine gland, which means that the secretion which is produced here by these cells travel through the blood circulation. So now you think about a situation where you have blood vasculature which is carrying these signals, and these signals were produced by cells here which are far off in the body. So signals were produced, they were taken up into the blood circulation, and at a far off distance we can say that there is another cell which has the receptor for this signal which is traveling. So basically you will see that depending on the messenger travel, whether it is traveling to a shorter distance or to a larger distance, you can have a short range signaling, which is often seen in autocrine signaling or in exocrine signaling, or the messenger is traveling to a larger distance, which is often seen in the endocrine signaling or paracrine signaling. Paracrine signal is nothing but the cell which is producing the signal has a receiver cell little far off, which is not self-type. So another way to depict this paracrine signaling, if I can just draw here and show it to you, is that you will have a cell which will produce the signal. So if you consider one cell with its nucleus here and these secretions produced, it comes out, it will travel to another cell. This is cell A. Now you have cell B, which is here, which should have the receptor for the signal, binds here and responds. Then you can have another cell which is at little far off distance, which will signal to this and go. Suppose this is cell C and this is cell D, which also has the receptor for that. So basically what is happening here is that you will be producing the signal which will be traveling certain distance to ensure that signaling aspects are met. Now there are very specific type of signaling which would be one of those types which we have discussed earlier, and this is where the signaling which is called synaptic signaling comes into play. You all know what synapse is. Generally two neurons which come together and they form a synapse structure. They come in close proximity, right? So if you consider this as a neuron cell, the action potential travels and allows the release of neurotransmitter in this small space which is called synapse or synaptic cleft. And then you have a cell here which is going to respond to these signals. They are binding to these receptors, right? So I told you that synapse generally we understand in case of neuron to neuron, right? But these neurons do form synapse with non-neuronal structure. It could be sometimes a muscle, where the synapse are referred as neuromuscular junction. Or these neurons can be terminating or forming the synapse with cells in a gland, which will basically give signal to these cells to make secretion. Like when you salivate, basically neurons send signal to the cells in your salivary gland to produce saliva, right? Similarly, the hormones which are produced in neuronal structure, you probably understand that brain has two distinct neuronal structures which are hypothalamus and pituitary, which are neuronal in origin and nature, and they produce hormones. So over there neurons are producing hormones and they are working on to the different cells. So the synapse where one neuron is forming another junction with another neuron or it with the other cell type. Here it is a classical example. If you see in this case, it is a short range signaling, right? Now what else can happen with the signaling is that when it comes to the long range signaling, I have already told you, I am trying to explain this in context of the endocrine signal. So I already drew this image. Over there we are trying to understand the long range signaling. Basically same thing applies here. You have an endocrine cell which will make secretion. It will travel into the blood vasculature and travel to a distance where you will have a target cell which will bind to these signals. These receptors will bind to the signal and do the signaling. Remember this as a general rule of thumb, that the cell which bears the receptor is the one which is signaling, right? So we saw different types of signaling which is happening, whether it is long range signaling, autocrine signaling, paracrine signaling, or endocrine and exocrine type of signaling. Now when we talk about the receptors which are involved in sensing these ligands, they will also drive the way signaling will look like, or the type of signaling which we are understanding in this class could be driven by the way receptors are presented, right? So these receptors could be a soluble entity or a membrane bound entity. When I say soluble, what do I mean by that? That the receptors are not bound to the membrane, rather they are present in cytosol, free from membrane, right? In that case, what we would notice that somewhere this is your target cell. Now I said consider the situation where it is a soluble type of receptor. So what does it mean? That it is not present on the cell surface? No, this is not the scenario. The receptor is not present on the cell surface, rather it is present inside the cell. It could be present in the cytosol or it could be present in the nucleus. For this purpose, what you need your signal to do to come inside the cell. When your receptor is soluble type, not present on the cell surface, you would need the signal to travel inside the cell. If you remember in earlier class, I told signaling could be considered like a bacterial transduction, where bacteriophage was binding outside, not coming inside, but injecting things inside. You can consider that this is a situation where this signal is actually going inside. Once this comes inside, it will find its receptor and bind to it. Sometimes the receptor has the function in the cytosol and performs the function. Sometimes these receptor and signal complex travel inside the nucleus and they do the job of transcription. Okay, so basically you will have one scenario where receptor is soluble, it must find its ligand inside the cell, so the signal will travel inside the cell, it will bind to it and then mediate the process either in the cytosol or when it goes to the nucleus, it may mediate the process of transcription. What is the other scenario when the receptor is membrane bound? Basically what we have been discussing until now is the membrane bound type of signaling, but let me come back to soluble receptor once more before I go to the membrane bound signal receptors. That the property of ligand which can actually cross this lipid bilayer, it has to be hydrophobic or lipid soluble. Right, you agree with that? Because if it has to enter inside the cell, it must cross this lipid bilayer. So this signal should have the hydrophobic property or the lipid soluble property. But the environment outside the cell is not hydrophobic, rather it is rich in water, hydrophilic environment. So such signal, they need a cover or a carrier molecule. So for soluble receptor, the ligands often are hydrophobic. To remain functional in the hydrophilic environment outside the cell, these signals, they need a carrier. So if I can draw this aspect little more clearly, it would probably look like this, that you have the, suppose this is your signal, you will need something which protects it from the outer environment, and this is nothing but your carrier protein. Okay, so this will allow the entry of the molecule inside the cell. And I have been telling you about the signaling and the presence of receptor on the target cell. That is basically a classic example of membrane bound receptor. So what will happen in such cases is that you have the cell, you have the nucleus, you have receptor, receptor will probably bind to its ligand. In this case, signal need not travel inside the cell. Rather, it will bind efficiently with the receptor on its surface and it will activate the signaling. Like we saw in previous case, the signaling could mean that changes happen in cytosol, or it could mean that the signaling culminates inside the nucleus and more transcription happens. We may later further take into account that the receptor may bind to the ligand with a poor affinity and mount a response, or there is a different receptor which binds with the ligand much tightly and generate a different response. Okay, so basically what we have learnt in today's class is that signaling type varies significantly, and we can define it in simpler ways, that the signaling could be between cells or it could be within the cell. So it could be intercellular or intracellular signaling. Depending on where the source cell is located, which is generating the signal, and how far the signal is traveling to find the target cell, it could be short range signaling or long range signaling. Further, whether the secretion is going into the proximity and working in autocrine manner, or the ligand is not free as a signal which is outside, rather it is membrane bound. When both the receptor and the ligand are membrane bound, you probably activate the contact mediated signaling. And the final thing is that whether your receptor is soluble or membrane bound, that also changes the way signaling will be activated and the downstream effects of that signaling. So thank you very much for this.